Okay, so guys, I just wanted to go through the just kind of the remind you of the content. And these slides, as Vanessa mentioned, are on the GitHub page, the full slides, because I'm not showing the full presentations. But so I'll just mention again the basic SCT data types that we create and upload under MONet. Then quickly go through the content of the previous two working group meetings, XCT working groups. I show that GitHub that was mentioned, and then I'll give it to Janshu. So just to remind you, on your cores, we do 4 scans. 2 scans are kind of mapping out the whole core. These are merged into one image to show the whole core image. This is lower resolution. We do not use it for the porosity calculation or analysis in Aviso because a lot of that porosity content is lost at this resolution. So for that we collect 2 more scans as shown on the right, top and bottom, basically halves, and that's where we do this full analysis of the total porosity, pore connectivity, pore network, and create these numerical values of the pore sizes and the distribution. And we also run this so-called pore network modeling module on it that is able to estimate flow and transport properties, and shows the so basically using the connected pore network, it can estimate permeability and flow tortuosity. So the image data is one part of what we provide. This includes the whole core, and then those different saved images of the segmentation work, that the higher resolution sections that what I mentioned for top and bottom, and then also we also upload an image stack for each porosity, meaning the connected pores, unconnected pores, and all the pores. So basically 3 image stacks are also added that can be opened, and so you can actually visualize the pores for yourself. And then the numerical data are summarized here by Billy. Just you can look at this in detail. Just basically points out each quantity or value, where is it found in the spreadsheet. And Janshu will give an example of how she uses such spreadsheets for her calculations. Oh, this just stayed here because we will put these on Science Central. The CT data is not there yet, but it will be part of it. So it can be accessed by sites and so from the map. And this is the GitHub where you can see the working groups, working group meetings, and we can upload the presentations here. So in the first meeting, I gave an introduction to the principles of XCT imaging, and this is a picture of the EMSL scanner that we use for those scans of your cores. Again, a summary of the 2 different type of scans that we do. The higher resolution scans are 38.2 micron voxel size. That's about 75 micron-ish actual spatial resolution. And then I also mentioned some other data formats that are not created as part of the standardized analysis, but we can. So upon request, and that's true in general, whatever you would like to do with the CT data for a publication or your work, reach out to us and we can work with you to create the right images and or data format conversion. So these are what I listed here are not currently part of the what we upload, but can be easily obtained. And then in the second meeting, Maruti gave an overview of some of the uses of the CT data. how to integrate it into his workflows for modeling. Basically, he showed examples of segmentation of the data that we use. We use Aviso for that in the MONet project, but the data can also be segmented using other tools, and he showed an example of how to do that segmentation using Google Colab, and then using, instead of Aviso, a freely available visualization tool, ParaView, to show the segmentation in 3D, and then he showed how the data can be input into PFlowTran for basically physical flow modeling, and he mentioned some applications of that. Basically, the goal is to provide this type of tool for users to simulate multi-phase flow with chemistry in the end. So basically, having the chemical information in 3D and in time would be possible using CT data and the PFlowTran workflow that Maruti has been developing. And I think Brian will mention some examples of flow experiments as well today. These are just again, these slides are available, just the key takeaways that Maruti summarized with the GitHub pages and links for to the software last time. And yeah, I want to thank the whole MONet team. And with this, if you have a quick question, let us know, but I will unshare and let Janshu start her presentation. Thank you.
Thank you, Thomas. Let me share my screen. Let me try. I'm sharing, right?
Yes. Oh, we see not the presenter view yet. Okay, great.
It should be working, right? Okay, awesome. Well, glad to be here. My name is Jianqiu Zheng. I am an earth scientist from PNNL as well. I'm not on the EMSL or MONet team. Instead, I'm more like a user, right? So my expertise is in soil biogeochemistry and microbial processes. So a lot of my research interest kind of overlaps with all these techniques that I'm just been providing. So I have been working with Thomas for quite a while. We're using XCT data for my own research. I'm just happy to share some of my inspirations and how I use this kind of data. So a lot of my interest comes to better understanding the complexity of soil microbial processes and build biogeochemical models for predictions. And as we all know, building models is complicated, especially for microbial processes in natural environment. These processes are different to model because beyond microbial diversity, that's whole other story. There are lots of complexities that are coming from the heterogeneity of the natural soil habitats, right? And the other aspect of this heterogeneity comes from a lot of those measurements are point measurements. You can characterize or measure a rate at a given soil sample at a given location and point, but for the ecosystem context, a lot of times we want to expand that understanding to a soil card, to even to a regional level. That's where we have a lot of upscaling. So that's where the processes have non-linearity when we think about upscaling and how there are emergent behaviors where multiple controls are at play when you scale up the understanding, right? So that's kind of my general research interest. Along that line, I want to give a simple example, like my favorite thing to model is soil moisture respiration relationship. Like we know soil respiration is so important, and there are a lot of drivers like temperature, moisture, they're typical ones. In theory, we have a pretty good understanding on moisture control on respiration. So microbial respiration will usually peak at intermediate moisture condition. So in theory, it was because that's the optimal moisture level that would allow a solute diffusion, which carries many carbon substrate, and gas diffusion, which carries oxygen for the reaction to occur. So that's the theoretical basis. If we look at real measurements from both controlled laboratory experiment and field data, it seems these observations are following similar pattern, either in the lab where they're more highly controlled and their temporal scale more likely the rate measurements are based on week-long incubations, or in the field where these are more instantaneous flux measurements based on flux towers, right? We can see that generally these observational points seems to follow similar bell-shaped curve. However, they're much more spreaded. So here in the graph with the color coding, I just used one of the many soil characteristics to show, across the clay content, actually, this respiration measurements points that they kind of really very well spreaded. And most importantly, the optimal moisture condition where the respiration rate would peak, it really varies quite a bit. So to be able to better understand these processes, I think our first action is trying to understand why these measurement points would spread around this theoretical curve, and how we can better constrain this kind of spread of observations. So I always start with the theory. Again, starting with the theory, I was thinking, okay, that's where the soil heterogeneity and the power scale processes comes to play. If we imagine a very simple system, you have a microbial cell that stays in this complex soil power network, you will have to have substrate diffuse into the reactive side that close to the microbial cell. That's where the reaction will occur. So eventually we can use very simple equations to describe this reaction. One is a power scale mass transport, diffusion, right? So the simplest one is use a linear diffusion flux. And then microbial kinetic reaction, simplest one, McCullough's mountain. So eventually, I think the reaction is controlled by this both of these processes, right? You have substrate diffuse into the reactive side, and that's where microbial kinetics process are going to occur that will finish this reaction. So we can actually solve the combination of these two equations to get a steady state concentration estimation. So that concentration is not going to be the same from the bulk soil concentration. It's going to be different because we have this kinetic term, we have this diffusion term. So eventually this expression shows the part of that is a kinetic control, part of that is diffusion control. It's a classic Dam-Kohler number kind of question, right? Is the diffusion more important? Is the reaction more important? Now I'll show you some of the results. These are theoretical considerations I started with. Well, think about the Dam-Kohler number. Depending upon how big is the Dam-Kohler number, the microbial reaction rate is either going to be linearly related with the concentration or not. Obviously, when it's a more diffusion limited system that we can describe, likely it's going to be linearly related. The diffusion is the rate limiting step of microbial reaction. However, if it's in a more homogeneous condition where Dam-Kohler number is really, really low, it's going to be kinetic reaction limited system. It's more like a nonlinear McCullough's mountain kind of kinetics will control the output, right? So now plotting this kind of equation, this theoretical understanding to the soil moisture kind of axis, we can see that the Dam-Kohler number would vary depending upon the moisture condition, also depending upon the diffusion length we have to use in this kind of theoretical calculations. Now, putting this even back to consider both carbon diffusion and oxygen diffusion together to create that bioshift curve, we can see that like subplot C, where these microbial reaction rate and moisture relationship, they show up at the bioshift curve, but how wide that maximum reaction rate would occur is really depending upon the diffusion length. That's where it goes back to what is diffusion length, right? So diffusion length is where it's really depending upon the soil pore network characteristics, how fast, how long the substrate have to travel to reach that reactive site. So based on this theoretical model, I did a numerical experiment, really thinking about not a unified diffusion length, but think about a distribution of diffusion lengths that kind of more realistic in terms of heterogeneous environment. If like here, I think I used like 1,000 diffusion lengths. They follow a distribution curve, and we drop them and think about their reaction rate, moisture curves. I would get this curve on the bottom right. So eventually, each diffusion length, they are coupled with a different kind of diffusion curve, reaction curve we can generate. So whether in terms of how large is the rate plateau would sustain, is really depending upon the diffusion length. So the nice conclusion of this kind of exercise, it gives us this idea that soils are inherently heterogeneous. If we think about how to doing a true spatially averaged reaction rate estimation, it's always going to be different from the main field prediction, meaning just using the mean diffusion length and the mean moisture condition to make the prediction, right? So that's the bulk-based predictions we usually make. They're always different. And more importantly, this spatially average behavior is always lower than the mean estimation. So this is actually could provide this kind of explanation why, yes, the theory works, but when you across different soils, across different sites, across like different measurements, they tend to vary, right? That they tend to diverge from the curve because we're always trying to do this bulk-based estimations. So now this is all like research institute I have in terms of, why I care about soil structure. This is all linked to soil structure. How could I get this diffusion length measurements estimation, right? That goes back to the XCT. That's where I know Tomas and his team has been doing this, sending this XCT scans, and then they provide users with this kind of images and the information on the pore network. In particular, and he mentioned these spreadsheet where they reported the geometry of the network, and they also report some like very detailed information on the soil pore distribution, pore throat, and the connectivity information. So for me, taking those spreadsheets, those information, I was able to use pore network modeling to create really cool visualizations first, and then really trying to think about what it means for this heterogeneity in soils, right? So here I'm just showing an example data set from Tomas. This is a Whidbey Island. It's a wetland soil. You can see that from my 3D pore network visualization on the right. So the soil pore network are pretty connected, but these pore sizes are pretty spreadly distributed, right? So they're kind of fluffy, and very connected. This is the kind of information I use from XCT. so coming back to think about pore network, what it means for pore network modeling. I don't know if maybe Brian may talk about this, about CFD kind of solver, think about more realistic numerical representation of the pore network that we have to solve for flow transport. I think about it in a simple way, right? Pore network modeling is really thinking about this soil pore topology, how we can just take that information and abstract that into idealized system where we just use different pore bodies, those circles, and then parse road, and then connect them, right? So they're very abstract. And hoping the idea is that they are very simple and abstract, but they should contain the same information in terms of the pore network characteristics. So ideally, this kind of information would still be very useful when we do calculations, yet because it's idealized, simplified, computational-wise, it's more efficient. So that's the whole idea, using the pore network modeling instead of real kind of numerical simulations on that. So just to give you a taste. So here, like the pore network modeling results, these are from different soils. We can see that actually different soils have very different pore networks, right? So just based on the visualization, like the process soil, the wild or the natural process soil is a grassland, can see that their pores are much more smaller comparing to the Whitby Island. Oh, there's the figure got cut. The Whitby Island is in the bottom middle. Like they are more fluffy, seems that they have much larger like pores. So maybe I think my hope is that, oh, maybe this pore network modeling, this simplified pore network could provide me enough information for what I want to understand. Different soils have different structures, and somehow they would reflect back to the soil function, maybe microbial respiration, right? So the calculation is very simple because it's a very abstract system. We just think about two soil parts that are connected. They may have different radius, and they are connected maybe by a different throat. So eventually, for the diffusion, we would assume most of these microbial activities are occurring in this, in the soil parts where there's because they have that kind of ideal volume for like microbial colonization and for more kind of microbial interactions to occur, are very simplified. So we can actually start to calculate the diffusion lengths based on the pore network features, where and start to plot out the distributions of that diffusion lengths. So eventually, this information we should use the information to feed back to this theoretical model, where the distribution of that diffusion means we can recreate that distribution in this numerical experiment and trying to think what it means for soil respiration and what it means for different soils. So here, like the different columns represent a different soil, like a clay-rich soil, a sandy-rich soil kind of situation. So that's my way, one of the ways that I can use this pore network information for these kind of theoretical testing. I have some of those ongoing projects that are using similar information. So here I just want to give you an example. This is a DOE ESS University award, where we're really thinking about whether we can use the differences in soil pore networks to explain some of the greenhouse gas flux measurements we made at like using the chambers, right? So the nice thing about this kind of design is that we actually have upland transition, wetland gradient, where sure their moisture are different, but likely their pore network and their pore connectivity are drastically different as well. So we are hypothesizing that these kind of differences in the pore network, like the throat diameter, the pore diameter, and they would control the diffusion processes, and eventually these controls should reflect on the surface flux measurements. So here, just with the first peak, like upland soil, it seems they have much complex soil pore network. And it seems we were hoping that this may help us to explain some of these patterns in the CO2 emissions. These are just small, like, ongoing projects we're doing. So if you want to know more about what I have been talking about in terms of the approach, here are two of my recent papers that that shows the details in the calculations and showed how that can be used for theoretical consideration of how microscale heterogeneity would control microscopic soil respiration patterns. That's all I have.
Thank you, Jenchu, and I'm trying to, well, if I can ping you later to get the titles of your papers so I can put them in the notes. Now, if people have questions, we can now transition to some of the discussion. Feel free to unmute. I think everyone can unmute or put your question in the chat. While people get warm up, I have a question for you, Jenchu. So if I follow everything correctly, you seem to characterize the pore network once in the soil and then use that in your model. Do you have a way to address changes in the pore network as a function of inundation or soil moisture? You know, kind of like if you're like more than just a snapshot of pore network. If there was like a system that was perturbed a lot or virally inundated, could you simulate those changes in the pore network within your model as well if you have one snapshot?
Yes. So the snapshot I take is just a pore network structure, right? Usually they got scanned at this natural moisture condition, but in the simulation we can actually make assumptions about relative saturation and then to give it probabilities or percentages that are saturated, so we can reduce the network connectivity because the soil is drier, or increase the pore network connectivity because soil is wetter. We can play those numerical experiment. But I kind of like, I was just thinking, because you're talking about one snapshot, I think that's the challenge I had in terms of directly using pore network data to support some of those modeling experiments I have been doing, right? Is how do you justify? Because these CT scans are segments of soil cores. They're each section, they're different, right? So do you average them out? How do you capture that characteristics? I think that's pretty challenging. It's like you want to be able to capture the variability within that type of soil, but with different type of soil, you kind of hope they're drastically different. So you can make explanations. I think that's been a challenge.
Yeah, and you know, before I pass it to Tamas, I wonder if there would be like a way, you know, I don't know if this could be possible, but that you have the core and then you disturb it somehow and then rescan it or kind of do some more like real-time disturbance to the core as you're scanning. That's obviously beyond my field, but I think it would be cool if something like that could be possible. Yeah, Tamas, sorry.
I'm going to chime in. I totally agree. Actually, one of my DOE projects, we're trying to do that, like doing a couple of indonation cycles and then try to rescan the same car when they're disturbed. But operationally, it's really challenging. So currently, Kaisa we have a scientist here who have a small project who's trying to do the freeze thaw cycle. It's a similar idea. I was just discussing with him about how on the modeling side, what I need, right? The idea is using the same car, and then you adjust the moisture condition, you freeze it, and then you scan again. Like, imagine that will maybe kind of enlarge some of those parts because they were frozen and then with the water, right? But operationally, again, it's challenging because we often want to do some other measurements as well, but then the car has to be intact. It sounds like we have to absolutely use the same car, right? So that's what we're trying. So it's going. Tomas.
Yeah, so Janshu, what do you think of the idea of doing a kind of a preliminary model study of your modeling, using MONet data that would just put it out there, show the community that that this can be done with this type of data?
Oh yeah, that's totally on my to-do list. Like
Okay.
I think eventually MONet data may kind of provide a soil catalog. Like we have the soil information, we have soil grid database, they tell you a lot of information on soil. But what about the structure? Maybe from the MONet, we can start to put them in, different soil textures, they tend to be associated with this kind of pore network, right? Or their levels of connectivity or their pore size distribution. I think that's where we can start to really think about this kind of soil structure data, how much value they can provide for general like biogeochemical modeling. So now I'm using distributions. I think they're useful, but at the same time, I'm challenged by a lot of soils, the distribution of the diffusion links, the pores, may not fully capture the property of that soil structure, right? It's a simplification. But when it's a simplification, you lose the information. So yes, in my model, I reproduce that distribution curve. That's why probability distribution function, because it's helpful for scaling, where you can expand your results from one soil car to your ecosystem, to your whole soil. But the downside is like when you do this kind of scaling simplification, you lose some details. So it's a trade-off.
Yeah, thank you.
Thanks. Hi, Brian.
Hey, Jianqiu. Yeah, so I think we discussed this before. So in this reaction network, like respiration, you have this pore networks defined in your model. So I think one of the question is, what is this major or characteristics lens that is playing the key role in those reactions? So I'm not wondering if you have done anything further on this line, just to identify what is the let's say, most important length scale associated with this particular processes that you are interested in, just based on the pore sizes or other things. Yeah.
That's a really good question. I have done a lot of literature search to think about what is the pore size distribution that are most important for microbial activity, right? It's around 100 micron, either based on soil physics estimation or based on microbial cell counts in soils, how the kind of the volume they interact with each other. There are some studies on those. I sort of settled at 100 micron. But on the other hand, I think a lot of this understanding are limited by how high of the resolution we can go. A lot of times,
Yeah,
right?
I totally agree with you.
Previously, the XCT, they're pretty coarse resolution. They do not resolve the fine parts or like that were isolated, disconnected from others. Are they important? I know their studies were showing there are carbon substrate in those isolated small parts, and they're actually yummy stuff. So they can potentially be very important when you have a major disturbance event, right? But a lot of times using XCT, you can't see it. But I know XCT has been improving with time. Also, there's moisture retention curves that potentially give you the whole continuous distribution of pore sizes. But again, this is a question, I think that's a very important question that we don't have a good answer yet. We have some understanding. I feel that's where this part-to-chip kind of work with the Terraform may provide an experimental platform to explore some of this hypothesis.
Yeah, I agree with you. Thank you for the answer.
Thank you.
Can I ask a question? I don't know why I raise the hand and it goes away. But I was going to give a chance to Marcus or Charlie if you guys have any question, because I can reach and find Jianqiu anytime I want for my question. I don't want to take your time out of that. Are you good? Marcus or Charlie, you have a question? Okay.
No, go ahead, Odeta. Go ahead.
Yeah, no, Jancho, one thing that I was trying to understand is the diffusion model that you said.
Yeah.
And a couple things with that. I've looked at the diffusion from, different perspectives, contaminant or maybe liquid movement or aqueous movement, right? In the case of the microbes, why is diffusion so important? Microbes are big, right? They are 1 micron or larger. And the diffusion, especially when you talked about clay, we're talking of very, very small pore space. So why would be the diffusion important in those scenarios? Microbes are not going to get there anyway. I mean, there's no space. So why would you care about the diffusion at that level?
That is a really good question. So the diffusion, obviously the diffusion I've been thinking about are the solute and the gas, right? So microbes are there and the diffusion, I think this is more from the perspective that the concentration we measure or infer from a bulk soil sample is not the actual concentration the microbial cell would experience. Their environment that at the micro scale is very different from the bulk measurements. So I was just trying to use that diffusion model to bridge that difference in the concentration. So that's why it's very theoretical. It's very simple, just a linear way, but that's a way to address that concentration difference around a microbial cell versus a bulk soil measurements.
Yeah. Okay, great. Thanks. I don't want to take more time.
Any last questions for Gen 2 before we move on? Okay,
Just
Billy.
quick comment. Genshu, you were mentioning Kaizad's experiment with the freezing and refreezing, and then the difficulties with re-imaging. We actually just developed, or I did, I 3D printed a thing that will actually it'll lock it in place. So we can actually do 2 identical images only if we scan the first one and then this, like, it sounds weird to say it this way, scan the first one and the second one, but we have to put this thing on it and we can do that now. 3D printers are awesome. I interrupt the comment, your pore network model is better than mine. I'm just saying, it's way better.
I just took your data sheet and kind of recreated that. But
No. Pat yourself on the back. Yours is way better.
So again, I'm more of a user, but I'm happy to discuss like how to use this kind of data for soil research. That's kind of my happy place.
Okay, great. So Janchu, if you're okay, I'll add your email to the note sheet in case other folks want to reach you later. And I'll also put the link to your papers along with the notes. And then, we can stay tuned about what other cool research you're doing. And okay, well, thanks again, Janchu. So now I'll pass it to Army and Brian for the next presentation.
Sure, yeah, I'm gonna share my screen really quick and I'll go into presentation mode in a second. Okay, how does this look?
Great.
Okay, awesome. Yeah, thank you, Genshu. I just wanted to take this moment to talk a bit more about Portachip, and I'm going to be presenting with Brian. This is a relatively new tool that we developed under the Terraform's platform for making reduced complexity 2-dimensional micromodels of 3-dimensional data. So we have a quick outline. It may not fit this time schedule. I'm going to try to go.
Do you have something covering the top part of your text? Because I see like a gray box. Maybe can you move it?
Oh, I see. That's the zoom bar.
Okay,
That's
great.
what
Now I
happens
can
when
see
you're
everything. Yes.
switching between Teams and Zoom. It's a nightmare. Okay. Yeah, so this is an outline about what I will be talking about. I'll be going over the introduction and how to use PortaChip and what it's capable of. And then later on, Brian will talk a little bit more about applications in flow and transport models and future applications with physics-informed machine learning simulations. And so this is the entire team that helped get PortaChip on its feet and up and running. Brian and I are on the left, and on the right is a team of people, including P&L workers and Aaron Rooney at USDA that helped through the user proposal to have this software developed. And I'll do a brief introduction of myself, and then Brian can go. I am Aaron Truong. I'm a post-bachelor here at P&L, and I work more on the computational side under Tomas's management for the terrestrial and atmospheric program here. And I am one of the lead developers for PortaChip. Brian?
Yes, so I'm Brian He. So I'm a mechanical engineer in P&L. So my PhD background is on the computational fluid dynamics, and after came to P&L, I started to do subsurface transport modeling. So basically, I'm quite familiar with both CFD and continuous phase modeling. Yeah, thank you.
Awesome. And I just want to give really quick credence and a shout out to Marty Mudanudo and Lal Mahmood. They are also co-developers for PortaChip. Lal being more on the chip to flow side, which we may have time to talk about a little bit later. So what is Terraforms? For those who don't know, it's a general platform for making higher resolution, reduced complexity platforms for observing different biogeochemical processes in soil aggregates or soil cores. And that involves, as you saw with the data that Billy Peterson can get in the XCT lab, the soil aggregate morphology, the porosity, but we can also get information like the mineralogy, the plants and their roots, and the microbes that grow in these soils and reproduce them in these lower complexity platforms called rhizochips in terraforms. And one of the reasons why this was the development of PortaChip was interesting is because Aaron Rooney wanted to be able to visualize and set a pore structure in a 2D micromodel that matched a soil core that was before disturbance and after disturbance. So she did some research using some soil aggregates of soil that had been not disturbed and soil that had been freeze thawed, and had a look at the pore diameters, the pore throat diameter ranges, and the coordination number, which is the number of pores another pore is connected to. This basically is one way to describe the connectivity of a pore network. And what she found was there was a change in the range of the pore throat diameters and also an increase in singly connected pores in the pore network after freeze thaw disturbance. And as Janshu mentioned, this could possibly be a big mediator, the structure of the pore network on biogeochemical processes, where if the water retention ability of a soil is increased or decreased depending on the disturbance of the soil, how will that affect the hotspots of microbial activity in different parts of the soil or the soil aggregates? An example would be if we had ash and sediment input into an undisturbed core, and this will disaggregate and cause a lot of closed off isolated pores and hotspots for microbial activity. And we want to be able to capture this in a low resolution or a low complexity micromodel without having to destroy our original core in doing any of these experiments, whether it be growing microbes or plants or doing medium flow simulations. And so from that idea came PortaChip, which is a way to apply a lot of computational and data analysis tools to XCT scans of soil cores and then build a micro model with the pore structure in mind when creating the platform. It'll look something like the bottom right image where you could grow plants or soils in it. So if you submit a soil core for MONet, you can have Billy do all of the analysis and get you the information that you want, including the segmented pictures of the XCT scans, and then you can use PortaChip to make actual physical micromodels for experiments.
Rami,
So,
may I ask, is this
sure.
transparent? Can you put this under a fluorescent microscope?
Oh yeah, absolutely. We'll talk more about the chips later when we talk about manufacturing. But yes, these are very transparent, and you can do a lot of your chemical imaging or whatever optical imaging on the chip afterwards. But so what is PortaChip? At its base, it is a Python module that we developed that will take the raw 3D scans of the XCT cores, and it can turn that, for a simpler way of saying it, into the 2D micromodel design that you would use for your experiments. But I say here that PortaChip is more of a workflow than just a software, because you can use a lot of different data types or different sources of data to get a 2D micromodel. So if you wanted to start from raw XCT data and you want to do this analysis on your own computing resources, you can, and you can get to a good place in that. Or you could have that data be computed by the MONet team and have all of your pore throat distributions, your pore size distributions and connectivity numbers from them, and then have us build a micromodel that way. It's a very robust workflow. And in our MODEX loop, it mainly, the PortaChip part covers this top part of our MODEX loop where we get our 3D pore network, we characterize it with the information that we want, build our two-dimensional mask, so to speak, and then create our physical micromodel to do our experiments with. Excuse me. Okay, and I want to talk really quick about if you want to get started with PortaChip, because all of the code is open source and it's available on our EMSL computing GitHub. It's all written in Python, so it's compatible with a lot of popular visualization and scripting Python environments. One of them that we use a lot is Jupyter Notebooks, but you could also run these in a Google Colab on Google servers if you don't have the computing power. And you could also run this on our supercomputing system at EMSL called Tahoma. So you can check the documentation on how to get access to Tahoma, and you can run a Jupyter Hub server where you run these scripting notebooks on this Jupyter server that's running in the supercomputing system through Open and On Demand. Some more technical details if you want to build or install it on your computer. This may take a little bit more coding knowledge if you like using the command line. To install it from the Python repository, you can just type pip install PortaChip. This will pull from the stable version of PortaChip we have on the Python package index. Or if you want all of the developmental features we have going, we can build from source. So you would clone it from GitHub on the EMSL computing GitHub site and then install it using pip that way. And another thing that we actually really recommend if you have Docker system on your computer is that you can build a Docker instance that has all the PortaChip dependencies installed with it and a small virtual environment that you can run your analyses in without having to deal with different OSs and installing Windows Linux subsystem necessarily. And to do that, you just follow these lines of code, build the Docker, and then just run it. And when you run it, it'll start a Jupyter Hub server with all the dependencies installed, and then you can play around with PortaChip. And here's the GitHub file structure for PortaChip. It might have changed a little bit, but the overall structure is the same. If you're super new to PortaChip, I highly recommend that you look at the example outputs and examples folders. It has a lot of the example micromodels that I'll be showing later in this presentation, as well as Jupyter notebooks that will get you at a high level from start to finish XCT data, raw XCT data to a micromodel design with markdown documentation all the way through so that you understand what Portachip is doing. If you're a developer and you want to contribute to Portachip in any way, we will be highly supportive of that. And to please look at the SRC folder. That's where all the source code is. So you can see all the scripts, what the functions are doing. It's all open source. And if you want to contribute to the test folder, so if you see bugs or you see features that could have some more code coverage in the test, they're all in this test folder here. Okay, so let's talk about the general workflow of Portachip. And before I start, throughout the rest of this presentation, if you see any micromodel or segmented image data that has black and white pixels, the black space will be considered the pore space and the white space is just the solid space, right? So from start to finish, how Portachip works is you have your raw image stack, so your 3D XCT data, and then we would threshold it or segment it into the pore space and the solid space using our built-in algorithms in Portachip. And then we would extract that three-dimensional representation network from this binarized image stack. And then we would extract the necessary information that we would need to make a micromodel, such as our pore size distribution. And then finally, we use our algorithm to reconstruct a two-dimensional version of that 3D model that you extracted. And if this looks familiar, this is the same thing that Jianshu showed earlier, except there's not really a visualization of the different pore sizes, but each of these red dots is a pore and each of the blue lines connecting them is a throat, and all of them have their respected diameters assigned to them. It's essentially like the digital twin of the segmented pore structure. And again, this is completely robust. If you had data that was, for example, extracted by Billy already, so let's say he already can give you the pore connectivity numbers or the pore size distributions, you don't have to go through all of these initial steps. You can go straight to making your micromodel. We're just providing tools so that if you started from nothing, you could get to a micromodel. Although we do highly recommend, if you submit a code, to have us do the image analysis, because we have an actual human that's doing the segmentation for us for the pore segmented images. Okay. And just a quick code snippet just to show you how compact and easy it is to get up and running. These are actual snippets from the Jupyter notebooks that are in our GitHub. So you could find these and run them yourself on your computer if you'd like, where all we do is we read in our raw data, use our filter function to filter the image, i.e. we're segmenting it and you can crop it or rescale it to whatever size you want, because I know XCT data can get very big. And then once we have this segmented 3D image stack, we can now input that into our metrics functions, which will return essentially all of the pore diameters, throat diameters, and the coordination number of every pore it detected in that 3D extracted network. And you see, we don't really see the 3D extracted network. It's all being done in the background thanks to these functions. And then when it outputs is a list of all of the radii of importance and the coordination number that I plotted here as a distribution for your reference. And this is what will be used to make the micromodel. Again, as I explained, the data extraction works using a watershed three-dimensional segmentation to get the 3D pore network. And this was helped by another open source Python package called PoreSpy. And like I mentioned before, for creating a micromodel with Portachip, the data of interest is going to be the pore sizes, the throat sizes, and the coordination numbers. And that's what we felt was going to be our best bet at characterizing the structure of the pore network in our 2D model. And I'm going to take a second to tell you what we're focusing on in the micromodel, which are the three things that I mentioned before, and what we're not necessarily focusing on. So one thing that we're not necessarily focusing on when we generate this geometry is the particle shape. And what I mean by that is the solid shape. So you might have seen some previous efforts to make heterogeneous soil geometries, and it often involved particle packing or sphere packing of solid shapes to simulate maybe a granular structure of soil. And the downside of that is that you don't really have control over the pore connectivity and the pore throat diameters necessarily. You can only change how the distance between each of the particulates that you're packing the spheres into. So the philosophy we had for porta chip is, well, instead of building it by packing solid material, we're going to build it by having a network that will simulate the pore structure, so how pores are connected. And you'll see that a little bit later, where the solid shapes are not necessarily our biggest concern. We want a geometry that shows us this is how the pores look, this is their diameters, this is how they connect, how many are connected to each other, etc. Another thing to mention that porta chip has the capability to do is to measure ferrite diameters of different pores. So for people who don't know, it's just a measurement of an object along a certain axis. So our ferrite diameter module in porta chip will return the maximum and the minimum values of a ferrite, so the longest axis in its length of a pore, which may be oblong, and the shortest one. This is, I think, important user metrics to have, or at least to see, and I plot it here as a distribution, but it will not be necessary when we're creating the micromodel. Another thing I want to talk about is the overall porosity. And what I mean by that is the percentage of pore space compared to solid space. We do have control over this, and I'll talk a little bit more about that later when we are constructing the micromodel. Okay, so here is where the porta chip algorithm shines the most. So let's say we have everything we need. We have our pore sizes, we have our throat sizes, we have our coordination numbers. Now we want to generate our two-dimensional network. And what we have here is a generate function in porta chip that will take in the diameters and the coordination numbers, and it will generate a micromodel or a two-dimensional network that matches the distributions that was given in these pore network parameters. So here is an example. This is a 10 by 10 pore network, and this is similar to that three-dimensional network I showed you earlier. It's essentially the same thing, only reduced in 2 dimensions. It's a reduced complexity now. And all of each of these individual red dots or the pores have their pore diameters assigned to them from the distribution that we extracted from the XCT image. Same for the throats, the blue lines, and it's same for the connections in between pores. That's where the coordination number comes to play. And what's good about this approach is that we're making this digital network easily and very scalable. So this was all 3 of these networks were generated with the same amount of data. It's not necessarily limited to, okay, I need a lot of data to make a big micromodel. No, it's very scalable. You can designate what the shape of your micromodel is. So if you want it to be 5 by 5 or 10 by 10 pores or 15 by 15, you can do that easily in the algorithm. And also, one thing to note, it doesn't have to be square. You can make rectangles for the purposes of demonstration. I made these squares for the presentation. Okay, so we have this two-dimensional network now, but now we want to make the actual geometry. This is what we're going to be using when we do our flow experiments, or if we fabricate them onto a chip. And what porta chip does is it takes this two-dimensional network, and then it will build a scalable vector image. In this case, it's an SVG image that will have each of the individual pores drawn as a shape with their designated pore diameter. And same for the throats and all the connections in between. So now we're using our 2D network as a scaffolding for this final geometry with all of the diameters accurate to what the 2D micromodel or 2D network had. And this SVG image, since it's a vector, it's very compatible with a lot of CAD softwares. So you could fabricate this through additive or subtractive manufacturing. In fact, on the bottom right is an image that I created in SolidWorks just as an example of this example micromodel, just to show that it could easily be done, and it's a very flexible file format. If you don't want to deal with SVGs, you could also export it as a DXF, which I know a lot of CAD softwares use. And it's at this point that I wanted to talk about how we're controlling the overall porosity of our final micromodel. So let's say, for example, we did our analysis on the XCT core, and we know that a percentage of that whole volume, let's say 50%, was pore space or empty space. And we want to capture that in the micromodel. So we want, in the micromodel, we want 50% to be pore space. And with this method, well, you don't control that necessarily. You can manipulate the generation terms to have 50% porosity. And what I mean by that is, let's say this image came out to be only 40% porosity. So one of the ways that you can increase the porosity to match what you saw in the three-dimensional XCT scan is to increase the number of pores in the overall micromodel. So maybe instead of 5, we'll do 6 by 6. So there are more pores, there's more density of pores, there will be more pore space, and that will increase your pore space. Another way is you could increase the overall size of the output micromodel. So if I say, let's say this is in pixels, I say pixels air quotes, but this image is in 50 by 50 pixels. If I increase this to 100 by 100 pixels and keep the number of pores the same, the structure will be the same, but now the distance between each pore is going to be enlarged. So there will be less porosity in that case. And so vice versa, if you decrease your size, your porosity is going to increase because now your pores are coming closer together, it's more dense. So while there's no parameter that say, hey, I want 45% porosity in this network, you can use all of our algorithms to manipulate our geometry in a way that will be most representative of your soil core. And again, this is a totally scalable process. Since these are vector images, if you zoom in, you won't see pixels or voxels. And I don't have units here because it is scaled when we did our analysis by pixels. But if you know the voxel size of your XCT scan, you can use that to scale your micromodel accordingly when you actually do your simulations or when you do your experiments on a chip. And here's an example that I was really excited to see, just a porta-chip in action, where this is an example from Erin Rooney's experiments where she took a normal soil core that had some water in it, and this was an aggregate that wasn't disturbed, and they did a freeze thawing of it to disturb the pore network, and then did another XCT scan, and these are the segmented images with the black pixels being the pore space. And this is where she saw the results of the more singly connected pores and more disconnected pores. And so we thought, what would happen if we ran pore-to-chip on these different aggregates? And you can see on the bottom left, this is a representation of the normal soil aggregate. And you can generally see it's a well-defined structure. There's a lot of connections, very few singly connected pores or lone pores. But now on the right, when we run the algorithm on the disturbed soil aggregate, we can see that there are a lot more lone pores, disconnected pores, singly connected pores. And this was really exciting to see that pore-to-chip was capturing these attributes of the three-dimensional pore network in a two-dimensional context. And okay, this is going to be about fabrication, which is one of my favorite parts. So on screen right now, we have a 3D print of the soil pores. I believe Billy did this one. Correct me if I'm wrong, Billy. And in front of that is an acrylic sheet that we used a laser etcher to etch our pore-to-chip design on. So this chip can be used now for your fluid experiments or to grow plants and microbes in. Now, we wouldn't actually use acrylic. We would use an epoxy resin slide, which I'll show later, but it is a very quick and efficient process using the laser etcher as compared to something like photolithography. For laser etching, it takes at most maybe 10 minutes for one micro model, and it's a very low maintenance, low headway and low labor cost, making it very fast and efficient to make. And it can get you details down to around 50 micron. You can go a little bit lower with some tuning, but 50 is usually where we say, okay, if you want anything, tighter than this with more tight designs or shapes than that, we would recommend photolithography, which is of course more time consuming, but it can get you micron details down to 1 micron, I believe. And here's a picture of one of our researchers, Arunima, holding it up just to show that it's real and not fake. But now let's talk about the actual slide and how it's made. So when we make these chips, we would start with usually a glass cover slip that could be coated with ITO for conductivity reasons, or it could not be. And then on top of this glass slide, we will build our habitat. This is where the micromodel design is, and this is the epoxy resin layer that is on top of the chip where the design will be. And this is usually, or this is made either by making the solid layer of epoxy and then using our laser to cut away the design to make our quote unquote pillars of the design, or we use photolithography to slowly build the layers up. And after that, we have usually a membrane, some sort of PDMS cover for when you do your experiments or flow experiments. And what's good about these is that you can now do your grow microbes and plants in these micro models. Whether you want to observe fungi mineral interaction or plant microbe interactions, you can do that now. And one thing that is not on the slide, but for this resin epoxy, you can actually embed minerals in it. And once you embed the minerals in the epoxy, whether it's photolithography or laser etching, we can expose those minerals to the air via one of these two processes so that microbes can interact with. Same with plants. So it doesn't have to just be only epoxy. We have ways of embedding mineralogy. There are, I believe, ways to scan the density of different minerals in your soil core of interest. So if you want a micromodel that has the chemical properties of that soil, we can definitely do that. And with this chip, since I know, Marcus, you asked if it was transparent, it is a fairly transparent resin and glass slide that you can do your optical imaging in, and all your biochemical assays and all of your chemical imaging in situ, so on the chip, and that includes things like MALDI, nanodeci, SIMS, all the technologies or the characterization technologies that EMSL has at our facility. So that was frankly a really brief overview of what PortaChip is capable of. I'm going to pass it on to Brian now, who's going to talk a little bit more about his CFD simulation. So I'm going to stop sharing screen really quick.
Okay, let me share my screen. Presentation mode. Can everyone see my screen?
Not yet.
Not yet.
I don't believe it's sharing.
Let's see. How about now?
Coming.
Yes.
I just have to switch to full slide mode.
Okay.
The other screen.
Yeah, the other screen.
Yeah.
Okay. Great. So what I'm going to present is some work that I've been doing recently So it can be seen as the extension of what Arami and other team members have accomplished. So basically, after we got those 2D representation of the pore structures or pore connectivities, how can we utilize that to do some modeling or simulation per se? So one of the options is to do this pore scale CFD type of modeling. So this slide shows the software that I'm using. I'm using this one called GeoChemForm. It is open source code developed by people from other universities. But it's the reason I choose this one is firstly it's open sourced, and another one is built on OpenForm. OpenForm is a very versatile numerical tool. It has all the capabilities that we need to do this kind of applications like meshing tool, solving tool, and other things. And also GeoChemForm specialized in this reactive transport and multi-phase, even heat transfer and some molecular level reactions. So that's what we exactly needed. Next slide is showing the workflow that I have built. So basically, I directly take image that Arami just showed. He generated this image, then he passed that to me. I port this image into the code, then let the code do the meshing. So here is a zoom in part showing the mesh of the actual computational domain. The grid resolution is about 10 micron, so it's a very small grid resolution. And then I'll let the code to solve for the Navier-Stokes equation to obtain the velocity and the pressure field at the pore level. So at each pore, you can see the very distinguished velocity and pressure distributions. And the way the flow is specified in this particular case is I give a constant 1 Pascal pressure gradient across in the x direction. So the flow is coming from the left side as inlet, then going through the domain to the right side as outlet. Next slide, you will see some more visualizations showing the streamlines of the flow field colored by the velocity magnitude. So one question I often get when presenting the CFD results is people say, what can this picture tell us? What is the physics behind those pictures except those colorful representations? Yeah, so that's a good question. I think I can talk about that. So from this image, we can see there is a main path of the flow from here into these pore structures and going through these different pores and throats, then leaving the domain from here and there. And that's basically the main path of any flow that can enter into these pore structures. But still, we have some very small velocity distributions in some isolate in the dead end pores, like here. The flow velocity is very small. So basically, this spatial heterogeneity of the pore structures directly controls this distribution of the flow field and also the pressure field. Another thing that is interesting to see is we see a lot of vortices that's kind of distinguish the flow structures in the domain. For example, in these pores and in these pores. So we saw those vortices in dead end pores, that's for sure, like this one. So because there's not too much flow going into here, so what's happening is most of the flow velocity is very small, and it generated this kind of vortices. But an interesting observation is if we see this pore, although it's an open large pore, some flow is passing through, it still has this kind of vortice generated. So basically, that is telling us even in this open single pore, there are two different kind of flow structures present. So that's kind of what this simulation is telling us. So even in a single pore, there's two different characteristic length scale or time scale associated. So the residence time for the flow and flow passing through this pore is much smaller for the residence time that is trapped in the vortices here. So those kind of vortices can significantly contribute to the long tailing behavior we often see in these breakthrough curves when we do experiments. So what that means is basically some, let's say, tracer or solute was trapped by those vortices in a single pores or dead end pores, which was released later on into the flow, then which shows as the long tailing of this of the breakthrough curve. So some other activities I'm doing now is basically computing this permeability based on the results of the computational simulations. Another thing is to do reactive transport, basically to add a tracer first, then we can simulate some simple reactions, dissolution, calcite reactions, for example. Yeah. And here are some current issues with this kind of simulation where I'm working on. First is this disconnected pores that Arami just mentioned as well in his presentations. So this on the left-hand side, I'm showing the original image, and the right-hand side is showing the actual computational domain. You can see those isolated pores are neglected in the computation because there's no way the code can compute any flow in those isolated pores. So we just neglected those intentionally. So computationally, there's no influence. But physically, if we neglect those pores, the total porosity will change, and which will again influence the calculation of permeability. So I guess that's an open question. So if anyone has any thoughts or suggestions, please can raise that up. The next question issue we are seeing is a convergence issue. For some runs, I have this converge, especially for the pressure term. It's very hard to converge. I refined the mesh multiple times just to. At first, I thought it's a meshing thing, but later on, I found it's actually the quality of the geometries. We see some sharp edge presenting in the geometry. So this is kind of the issue we typically want to minimize as much as possible in doing computational fluid dynamics, because that will raise issues to the convergence. So we can work on that later on. Yeah, that's the computation safety side of this modeling. So beyond that, what we can do is to use this physical informed neural networks. So that we call it PIN. So PIN is basically, you can see that's as a technique that can bypass the traditional CFD solver to solve for the whole flow field. It can directly solve for the partial differential equations if trained successfully. So this PIN, after we run a whole bunch of safety models, we can use that data as a training base to train this PIN network model. Then we can basically get a faster simulation results. Then after that, we can also use the results like the permeability we get from the safety models as input into P-Flowtran models. So for P-Flowtran, we need to define the soil properties at each grid cell. So that's where the porosity, permeability, and other things are needed for this kind of application. Then I guess Arami just covered from 1 to 4 for this whole workflow. Then I think Arunima is doing the number 5 here, doing experiments, and I'm doing the 6 pore scale simulation and maybe 6A and 6B. Then next step, we are going to this scaling technique, basically to scale the characteristics metrics that we have in the pore scale, then upscale into a larger scale to input into other code like P flow train. Yeah, so then we have this whole loop closed. Yeah, I think that's all we have. Yeah, if you have any questions, and Arami will be glad to answer.
Yeah, I also really want to say quickly, Brian, for the issue of the lone pores in the simulation. In ported chip, yes, you can generate a design that doesn't have lone pores. You know, for the sake of this presentation, I gave this one because it'll be consistent throughout the presentation.
Yeah. I understand that. It's just it's a computational issue.
Yeah. And for the meshing, that will be tricky.
Right. Yeah, but we can work on that just to, I think the one thing is to have a very smooth surface of these pores, then have a high resolution of the image that port into this solver should be able to solving this issue. Yeah.
Okay, I'm pretty sure, yeah, that could be solved since we export an SVG image. It'll be a very high resolution picture though.
Yeah.
I think we have a question from Charlie.
Yes, actually, I have 3 questions. The first one is, I'm wondering if there is a limitation for the micro model, like what is the smallest size you can make for the micro model? That's my first question.
In terms of the size, and it depends on which method you want to use. If you're talking about fabrication, so like on an actual chip, if you just wanted the two-dimensional design, it is totally scalable since it's digital. But when it comes to the actual physical chip, if you wanted, let's say maybe even as small as, let's say 3 by 3 millimeters, then we could make a micro model that is that small. It's just that at some point, if you want really high detail, then we would prefer to use photolithography, because a lot of the experimental chips we've been doing is with the laser etcher that can give us really fast results, but gets us up to or down to maybe around 50 micron pore sizes and pore shapes. Does that answer your question, Charlie?
Yeah, so yeah, that's kind of I think I also heard that you mentioned you could go down to 1 micrometer resolution if you want to
With
go higher.
photolithography.
Yes, so so it's like overall this very small micro model would still be representative of the like the core you started with micro CT, right?
Yes, and
Okay.
of course this also depends on the voxel size when you did your XCT scan. It'll also probably be different if you did micro CT, micro XCT.
Okay. Yeah. I also have 2 other questions. The second one is whether you have tested the like the physical properties of the micro model compared to the original core, whether they would match each other. And then I also wonder if you actually grow any plants in that micro model.
Well, I can answer the last one. We've grown microbes in these micro models before by putting them out in the field. I'm not sure about plants. I'm on the computational side and I'm on the fabrication, so I don't do many of the experiments, but we have, since PortaChip is part of Terraform's platform as a whole, there are rhizo chips that have grown, or we have grown plant roots in them, and PortaChip is really just an extension of here's another way we can make a rhizo chip. So I don't think we have done any plants grown in micromodels made by PortaChip, but I don't see why it couldn't be done.
Okay. Great.
And as for validating, and I think that's a really important question that it makes the whole MODEX loop connect. It is we want to validate the results from the two-dimensional physical chip with any maybe physics simulations we do in the 3D side, and that is still ongoing as far as I know.
Okay. Thank you. Sorry, I have to
Marcus?
sign off. I have another meeting. See you.
Okay,
Bye-bye.
thank you.
Thank you.
Yeah,
Very
and
good talk.
Marcus, you have a question?
Yes, but that's very interesting. Thank you for those presentations. That's really cool. It struck me when I looked at those images that Arami showed, you know, with the XCT, you have those cross sections, and then you make basically the mapping of the 3D structure to the 2D, and those images look very different, right? And that's because you have those targets, you know, you have the pore size, the throat size, and the coordination number, right, that you showed. And I think you need to go back more in the slides, maybe. One more. I think you had a slide where you showed those at the very beginning, I think it was. Here, yeah, this one, yeah.
Maybe I
Yeah,
passed
I
that.
believe it's the workflow one with the arrows is the one Marcus is referring to.
This one.
This one, yes, yeah.
Okay.
So if you look at the 2D cross sections right on the left, they look very different than the reconstructed medium that you have there. And so that's as I understand, the reason how that happens is that you have those three targets that you use, right? So the pore size, throat size, and coordination numbers, right? These are the three things that you probably optimize to create that,
Yes.
that 2D image out of the 3D image. So if I just look at those images, you know, they are very different, and you would expect very different flow through those two media. And that's probably not unrealistic because you are mapping a 3D to a 2D image, right? So that's very cool. I wonder why you don't include tortuosity in your targets as well.
Yeah, that was something that I had talked to my team about back when we were first prototyping PortaChip, and it is pretty complicated because how we're connecting and making this scaffolding, as you saw, for the two-dimensional network, we're essentially making a lattice that we can control the positions of the pores and their coordination number connectivity. So, and we really tried our best when we make that SVG image where the throats there, it's not just like a ball and cylinder model, right? The throats are of irregular shape, and there is a lot of development in the future for PortaChip, and I think that would be something that would be really cool to explore. I'm still working on a lot of different algorithms that will generate a good micro model, and I will say this image, I presented these images, I think back in June. The algorithm's constantly changing, and we're still working on PortaChip as a whole, as it's open source, and we really want a lot of developers to, you know, weigh in on this, on their different techniques. So I definitely see what you mean. And of
I
course,
think that what Charlie mentioned, you know, is with the validation, that is really interesting to do, because I guess, Brian, you can run those simulations also in 3D, right?
Yeah, I can. It's
So you
just a
could
demonstration of this capability.
right, yeah. So you
Yeah.
actually could validate it numerically whether the 3D version would match up. Let's say you just calculate the permeability, for instance, or a breakthrough curve of a tracer and compare the 3D and the 2D and see how well they actually match, right?
Yeah, I agree.
That's really cool. Yeah.
Yeah.
Very nice.
So one question, since you mentioned the tortuosity, do you have any other thoughts on other metrics that should be considered when doing the comparison between 2D and 3D?
Yeah, I think the tortuosity would be a good one. You know, that's of course this tortuosity is a more a continuum parameter compared with the ones that you use, right? But I think that would be important to do. The other thing I was wondering is what the impact is of the size, you know, in terms of representative volume that you do actually those segmentations on. Because you in Brian's, you had that simulation with these dead end pores, right? That you then, of course, you don't have any flow. But if you were to make the size bigger, those dead end pores may be connecting again, right? And then they would actually contribute into the flow.
Yes, I totally agree. So we actually have been discussing this with Maruti and other team members. So basically, that's a question of how to decide the REV for this whole domain. One possible way is to find the average length of each pore size or particle size. And based on that, we can decide the representative size for each segmented subdomain. But yeah, we have been discussing that, but haven't done anything on that yet.
Very nice. Yeah, no, that's cool. Yeah. Because I think that's a big advantage, you know. Certainly the cross section on the left is not representative, right? So
Right.
if you're going to the that 2D representative mode, that would be really great. Yeah, that's very, very cool. Yeah.
I had one question for you, Brian. Thanks
Okay.
for the presentation. That was pretty cool. First time I saw that, so
Thank
I
you.
really liked it. When you go to those isolated pores, and maybe also Marcus can chime in if he has any idea. I know that you were mentioning that how do you feel those, you know, doing this simulation? And I think in that case, I'm just going back to what Janshu said in the beginning about diffusion, because in a real system, that's when your diffusion is going to play a role to fill those isolated pores, depending, of course, from the moisture content you have in the soil, et cetera, et cetera. But I don't know how you can make that happening in a simulated form that you have right now. Is anything in the toolbox that can simulate the diffusion process here?
So basically, I think I understand your question. So basically, you want to see how the diffusion is impact to the whole flow field.
Yeah.
So for the safety type of simulation, we are actually solving the Navier-Stokes equation, which include this diffusion term on the right-hand side of the equation. But for this particular case, since we have very slow flow and almost stationary flow field, so on the left-hand side of the equation, you have this temporal terms, those advection terms, all the inertial terms should vanish numerically, inherently for this type of application. So what left is basically the pressure term and the diffusion term on the right-hand side of the equation. That's basically reduce the whole Navier-Stokes equation to the diffusion equation essentially. So yeah, so it is representing the diffusion in a numerical way. It's just it's neglecting the inertial terms when doing the computation. I hope that answers your question.
Yeah, it does. I don't fully understand it because I don't come from
It's
field that you
hard to
guys are, but
yeah.
I think I understand it enough.
Yeah, it's hard to explain. It's basically because except if I can write whole equation down and I can tell you which term can be neglected, which term is left. Yeah, that will be easier to explain that. Yeah, but I think we Jianqiu and I, we had some discussions on this as well. So basically, as the question I mentioned to her is how to identify the key correct length, which is contributing most to this all those interests in reactions. Let's say if reaction happening in those fast moving streams, so the characteristic timescale is really short. So anything goes into here will move fast, exiting the domain. But if some other things is trapped by the dead end pores or those isolated vortices here, so basically those things will remain longer in the domain. So it would have more time to react to do other things. Then that can change the whole behavior of the reaction networks.
Yeah.
So yeah, that's something we have been discussing, but yeah, maybe we can move forward on that.
Yeah, 100% on that. And that's why the chemistry is super important, especially on those domains that they, remain almost like a batch reactor, actually.
Right, exactly. So
for a very
yeah.
long time. Yeah.
Right. So we always think that the reactions are happening homogeneously inside of the pores, but essentially it's not. It's really spatially heterogeneous. So yeah, so that's a question, always a question for upscaling, how to have a reasonable upscaled result from the pore scale to the larger scale. Yeah.